Single cell sequencing is really important to understand the heterogeneity of cellular populations, but also to perceive how individual cells differ from one another at the molecular level.
Single cell RNA sequencing was and still is truly mind-blowing. It allows us to study individual cells even across millions of cells simultaneously.
When you think of single cell sequencing, accessibility usually isn't the term that comes to mind. Unfortunately, we couldn't do that many because the cost is prohibitive.
We knew how important it was going to be able to provide a complete end-to-end solution to support researchers with the analysis and interpretation of the complex data that can be generated with the Illumina Single Cell Prep solution.
Illumina's 3' single cell sequencing kit, which uses PIPseq technology, I think changes that paradigm. It's accessible and easy to use, which means you can do a lot more with it than you can with alternative single cell technologies. Setting up the Illumina single cell solution in the lab is really straightforward. You don't need any specialized equipment, just a heat bath and a vortex, and it's a really straightforward protocol. What surprised me the most about the workflow is how simple and straightforward it was. We got results without having to play around with the protocol.
Illumina Single Cell Prep is highly scalable with the same workflow and equipment. It supports a broad range of cell targets from as low as 200 to up to 1 million. It is super helpful for researchers who are working with limited input material or are aimed at capturing rare cell types that make up only 1% of samples.
Not only does it make single cell sequencing more accessible, but it also means that we can think about doing more samples, and that opens up new research questions. We can start thinking of doing population screening more accessibly than we would previously.
We have successfully generated libraries, and the traces look beautiful.
The quality of the data has been good. We're able to get the same conclusions we would with other technologies, except in a much more affordable manner.
It brings powerful, widely used analysis tools in a simple point-and-click interface with no coding or bioinformatics experience required. Now researchers can quickly explore complex data sets, uncover insights, and generate publication-ready figures, making high-quality science more accessible than ever.
Illumina's solution for single-cell sequencing has allowed us to incorporate this approach more frequently in our research. We're now thinking of doing single cell more regularly in complement to bulk methods.